السلام عليكم أعزائي الطلبة. اليوم راح نتكلم على موضوع صراحة شيق، وهو Immunoregulation. How the immune system will regulate by participation of the antibody, participation of the cytokines and T regulatory cells, also some genetic factors that involve in the regulation of the immune system. So the teaching objective is to discuss the regulation of the immune response, including regulation by antibody, T regulatory and cytokines. Second, to discuss some genetic factors influencing in immunoregulation. So the magnitude of the immune response is determined by the balance between the antigen-driven activation of lymphocytes and negative regulatory influence that prevent or dampen the response. Regulatory mechanism can act at the recognition, activation, or effector phase of the immune response. So in these topics, we will talk about the regulation by the antibody, regulation by the cytokines, regulation by the regulatory T cells, and the genetic factors influencing the immunoregulation. راح نشوف الفيديو يوضح تفاصيل بسيطة حول regulation of the immune system.
It's always a great honour to be asked to speak at this fantastic symposium, and I think up till now the lectures have been really superb. So I've been asked to talk about immune regulation, and I want to thank both Jintanat and Liddy for introducing a lot of the concepts that I'm going to deal with and putting them into context. So this is going to be a bit more of a general talk about immune regulation, because really, in terms of immune regulation, nothing is really ready for prime time in the management of HIV, whether it's to do with control of morbidities or immune reconstitution, or in the realm of cure research and control of latency. So I wanted to deal in the next half hour with what immune regulation is and why it's important, and how that then impacts on our designs of immunotherapies. I want to talk a little bit about how viruses actually affect immune regulation and actually go out of their way to dysregulate the immune system, and how they do that, but also how we might take advantage of those genes within within those viruses to come up with better vaccine design. And then I wanted to actually describe the complexity of immune regulation, to give you some background about how we would go about what the challenges are in designing rational therapeutics for immune regulation, effective immune regulation. So the immune system, as we all know, is a very complex beast, and for a lot of people, people are discouraged from studying the immune system because of its complexity. And in fact, much of its complexity is based around the regulatory components of the immune system. So it's very important we have effector mechanisms that clear pathogens from us. And they destroy pathogens. The immune system destroys pathogens and does that very effectively by killing them. The trouble is that there is always the possibility of collateral damage, and that collateral damage must be controlled. And so as much as the immune system, the effector systems of the immune system are important, it's equally important to be able to make sure that they're only turned on at the right time and they're turned off at the right time. And that's really what immunoregulation is about. And there are multiple checks and balances within the immune system that control the turning on of and turning off of those effector mechanisms. And they really can be summarized into control at two levels. You can prevent inappropriate immune activation, so you only turn it on when you really need to. And the best example of that is the role of thymic selection of T cells, so that you have only T cells that see foreign organisms and don't attack the host. There are other mechanisms such as peripheral tolerance, but the antigen specificity of T cells and B cells and the fact that you need more than antigen to turn on a T or a B cell effectively is equally important. So you need an antigen presented in a particular context by an antigen presenting cell, and that antigen presenting cell has to have cell-to-cell contact with the T cell or B cell and express particular regulatory molecules that deliver a second and a third signal that then turns on the full immune response. Lots of cells are covered by ligands that are negative regulators that actually suppress the function of those cells, and the sorts of NK cells that Liddy was talking about are covered with receptors called KIRs that make sure that those things are not turned on inappropriately. And even in the protein systems like the complement cascade, there are as many regulatory elements turning the complement cascade on as effector mechanisms within the complement cascade. So that prevents immune activation. The other thing we must do is we must turn off an activated immune system at the appropriate time. And this might be done by negative regulators such as PD-1, which Liddy talked about a little bit and I'll deal with a bit later, and other negative regulators like CTLA-4. The very act of antigen clearance is important in turning off an activated immune system. And then there are other ways of turning off an activated immune system, such as T regulatory cells. So the control of an immune response occurs at multiple levels. There are specialized tissues that control cell differentiation and cell proliferation. And within those tissues, the microenvironment in which these cells exist is extremely important. And in fact, there are some very precise control mechanisms that I'll show you in a moment. So those microenvironments control cell-to-cell interactions. And so the cells actually need to be cheek by jowl, bumping into each other so that specialized protein receptor ligand interactions can occur in a very coordinated way. These are further controlled by the levels of RNA and by the levels of microRNAs, and then further controlled at the DNA level by regulation of epigenetics. But this can be dysregulated by microbes, and particularly viruses have genes that allow them to hijack the immune system, to cause virally mediated immunosuppression, for the viruses to be silent to the immune system to allow them to avoid the immune system, and in other ways to mimic other proteins within the immune system. And of course, HIV is a past master at all of these, and I'll show you some examples in a moment of that. But other viruses also have genes that regulate these interactions. So if we just go back to the importance of tissues and tissue differentiation and the microenvironment, there are specialized tissues in which the immune responses are generated. So one of the best examples of this is the thymus, where T cells are made from thymocytes, and they mature in that environment. And they mature in a particular way so that you only get T cells that see foreign pathogens, and those that see self-antigens, host antigens, are deleted from the repertoire. So as these cells differentiate and generate their T cell receptors within the thymus, they circulate within the thymus in a very particular pattern, interacting sequentially with different cells, receiving different molecular signals from receptor ligand interactions that decide whether those cells exist or die by apoptosis. And this happens in a very precise, coordinated fashion within these particular microenvironments that are regulated by the stromal cells. And similarly, in the generation of a primary or secondary immune response, the main area where that occurs is within the lymph node. And again, this need for precise circulation of cells, particular interactions between T cells and B cells occur in particular areas of the lymph node. And there is this coordinated microcirculation that controls whether these cells interact or don't interact at the level of receptor-ligand interactions is very important for the generation of both the primary and secondary immune response. This is a cartoon showing the generation of the different flavors of T cells from a naive T cell. And so a naive T cell sees an antigen interacts with an antigen presenting cell that presents antigen in the context of MHC1 or MHC2. And then there are these particular receptors like CD28 that provide the second signal that then, in the context of the cytokines produced by the antigen presenting cells, determine by the microenvironment which sort of flavor T cell is generated from this interaction, whether that be a Th1 T cell, a Th2 T cell, a Th17 T cell, or a T regulatory cell. And so this cell-cell interaction, this close interaction in a particular microenvironment determines the fate of these cells. And similarly, interactions between T cells and B cells depend on a set of similar but different receptor-ligand interactions that require the cells to be in close proximity, and those determine whether memory B cells and plasma cells are produced. This depends on the presence, this generation of T effector cells and T memory cells depends on the presence of antigen. And so the antigen presented in the context of an antigen presenting T cell in the right microenvironment, you get proliferation of these naive T cells into a large pool of effector cells when the antigen is present. And these effector cells have different flavors or different colors that produce different cytokines in different ratios, like IL-2, TNF, interferon gamma, IL-4, IL-21. And there are also T regulatory cells that are antigen specific, produced at the same time to regulate this immune response. On clearance of the antigen, this immune response contracts. Those cells die by apoptosis and they disappear from the body. And from that population of effector cells, a small population of memory cells go on and exist for long periods of time after this phase of contraction that are maintained by various homeostatic interactions that include the need for IL-2 and IL-7. So I just want to illustrate how precise and elegant this interaction is within the lymph node, taking a lymph node environment as one example. And this is some experiments we did with our colleagues at the Garvan a couple of years ago. When we were looking at the role of T follicular helper cells and their circulation in a primary immune response, that is the first time you see antigen, and in a secondary immune response or in a memory immune response. And we were looking just within the follicles within the lymph node and the behavior of the T follicular helper cells. So you might think that T follicular helper cells are a homogeneous population. And T follicular helper cells are very important for providing the help that allows memory B cells and plasma cells to be developed, and in fact induce somatic hypermutation, which is so essential for the generation of neutralizing antibodies, particularly if we're ever going to have a HIV vaccine based on neutralizing antibodies. So in a primary immune response, the follicular T helper cells stay within the lymph node, within the particular follicle and germinal center in which they're generated and do not migrate out. And this is regulated by the presence of a cytokine, EBI2, and their non-expression of that EBI2. And so they sit within a particular germinal center and don't migrate out at all. In a secondary immune response, this migratory pattern is completely different, even though these cells on their surface look exactly like a follicular T helper cell. And so these cells have now much more freedom and migrate much more widely within the follicle, but also to other follicles within the lymph node. Now, how this impacts on memory, we're still trying to understand, but it does impact, but where these interactions occur do then determine the nature of the B cell response that you generate. So doing similar sorts of experiments, we've identified two separate areas where these T and B cell interactions occur within the lymph node. One is within the periphery of the lymph node and one is in the germinal center. And if these T follicular helper cell B cell interactions occur within this peripheral region here, you get lots and lots of antibodies very, very quickly, but they're very, very low affinity, and they're from things called plasmablasts that have a very short half-life. So you don't get a sustained memory cell response. You get a short, sharp response that's quick and dirty. If you have the interaction between the T and B cells occurring within the germinal center, you get selection of high stringency antibodies that have high affinity generated by B cells that are more likely to become plasma cells that are long-lived and memory B cells. And so if we were trying to generate an immune response to a vaccine that had long-lasting immunogenicity, we would want this second sort of immune response rather than the first sort of immune response. So targeting antigens to generate this sort of immune response is what we should be aiming at. But you can see how we manipulate these interactions. We're still a long way from fully understanding. So how do viruses impact on these elegant systems that the immune system has generated to regulate itself? Well, if we take HIV and how it interacts with the immune system, well, the very fact that the envelope of HIV uses important immune ligands such as CD4, CCR5, and CXCR4 to get in is a way of immune hijacking. And furthermore, the use of CCR5 is very interesting. So CCR5 is upregulated very early in both a primary and a secondary immune response on T cells. Doesn't matter what sort of T cell it is, you upregulate CCR5. And that's the very molecule that the virus uses to get into these cells. So it's there, it uses a ligand expressed by the immune system to target cells that are about to expand and proliferate to give itself the perfect milieu to grow in, but also to kill those cells that are being generated against itself and other viral pathogens as they're being generated. So nullifying effective immune responses. And Liddy showed that great data showing that follicular T helper cells, which actually coordinate this response and generate those antibodies and generate those T cells that you need, are preferentially infected at this point. And that's because of the sorts of the ligands that HIV has chosen to interact with. But the virus does much more than that. It has direct lytic effects, but it also has pro-apoptotic effects from other proteins it expresses. Envelope, NEF, VPX, all can cause apoptosis of T cells. There are other proteins such as VPU, VIF, and TAT that interfere with the action of type 1 interferons, either directly or through the proteins that type 1 interferons produce, such as ISGs. And then these proteins also have a role in immune evasion, making the virus less detectable by the immune system. Liddy was talking about the importance of HLA1 or MHC1 and the determination of long-term non-progressors. Well, NEF and VPU downregulate MHC1 so that HIV antigens can't be presented effectively to the immune system. These cell-cell interactions that I talked about are determined by cell adhesion molecules such as ICAM1, and VPU downregulates the expression of ICAM1. So you can see that HIV has gone out of its way to make sure that it immunosuppresses the immune system in other ways and fiddles with immune regulation in other ways than simply killing CD4 T cells. But other viruses have done this as well, and have done this to a much greater extent than HIV. So herpes viruses are the classic here, and they are about 12 to 24 times larger in terms of genomic content than HIV. But a large part of these excess genes are aimed at dysregulating the immune system or making sure the virus is less well detected by the immune system. So there's a lot of genes within the various herpes viruses like CMV, like EBV, like HSV1 and HSV2 that inhibit class 1 presentation, that prevent that presentation through HLA1 or MHC1 that Liddy was talking about that's so important for CD8 T cells. And there are multiple ways that herpes viruses stop the presentation of antigen through MHC1. But they also interfere with presentation through MHC2, which is important for CD4 T cell responses. They also fiddle with dendritic cell and antigen-presenting cell maturation and activation. And then they also secrete mimics of immunosuppressive cytokines like IL-10. So they've spent a lot of time in their evolution determining ways to dysregulate the immune system. So while this is clearly to the advantage of the virus, if we manipulate these genes, we can also perhaps do better at vaccine design. And the vaccine that Liddy talked about briefly, the CMV-based vaccine that protects macaques through presentation, atypical presentation through HLA-E, the mechanism of that actually appears to be the non-expression of certain of those immunoregulatory genes within that particular CMV. And so if we understand these immunoregulatory genes within these viruses, we may be able to manipulate them to produce unexpected immune responses like the HLA-E-mediated CD8 T cell responses that appear to be protective in those macaques, and build novel techniques for vaccines. So it's all not just doom and gloom about how clever these viruses are. If we learn about these viruses, can manipulate these viruses to manipulate immune regulation for our own advantage. I want to talk about the persistence of antigen and how persistence of antigen dysregulates the immune system, because this is the case in non-treated HIV-infected patients. So this is the same cartoon that I showed you earlier, but we don't get antigen clearance here. We get continual presentation of antigen in the presence of antigen-presenting cells back to the immune system, and you get this expansion of effector cell populations. But the profile of cytokines that these produce change, and it's disruptive. But also these cells become less effective. They become exhausted. They upregulate negative regulatory factors like PD-1 and CTLA-4 and CD57, and they're chronically activated as judged by the molecules that you've all heard about in the past, like CD38 and HLA-DR. And you get out of this, because you don't get the effective contraction phase, you get disordered production of effective memory cells. And so this is what happens in HIV. You get these exhausted, expansion of these exhausted effector cells. But this happens from very early on. This is some old data from our lab in Sydney, showing that within weeks of infection, this is now comparing immune responses to vaccinia at a week to people at about day 14 post-infection with HIV, so acute seroconverters, untreated. And already within 14 days of infection, these people with HIV are producing much more cells with much more CTLA-4, that negative regulator of T cells, and much less IL-2 and much less and expressing much less IL-7 receptor than an effective immune response, such as the immune response to vaccinia. So this happens from the earliest points of the immune response to HIV, but then is exacerbated by this chronic antigen presentation. And there is further upregulation of CTLA-4, further downregulation of CD127, the IL-7 receptor, and overexpression of PD-1 that means that these cells are ineffective and exhausted. So what is the role of PD-1 and CTLA-4, these negative regulatory cells? What are these negative regulatory molecules? So this is an antigen-presenting cell, a dendritic cell. This is a CD4 T cell, but this could be a CD8 T cell. Normally, there are these molecules on the surface of activated dendritic cells called CD80 or B7 that interact with the co-stimulatory molecule CD28. This provides a second signal that activates T cells and causes them to mature or differentiate or proliferate. The CD80 can also interact with CTLA4, which is a molecule that looks very much like CD28, but instead of giving a positive signal, gives a negative signal to the CD4 T cells and suppresses the CD4 or CD8 T cells, preventing them from producing cytokines or proliferating. And PD1 interacts with a molecule called PD1L on DCs that has a similar effect of turning off and limiting the proliferation and differentiation of CD4 or CD8 T cells. And this is shown in some more detail here, where the interaction between CD80 or B7 and CD28 turns on the T cell or makes it cytotoxic. But if you have CTLA4, if instead this interaction is with CTLA4, you get decreased CTL function and decreased cytokine production. But there are now antibodies that we can give that block this interaction, that block the interaction of CTLA4 or PD1 or PD1L and stop this negative regulatory signal. So PD1 and CTLA4 are both increased in expression on CD8 and CD4 T cells. And as I've said, the T cells are exhausted and suboptimal. And PD1 expression increases with disease progression over time. So why don't we just block these molecules and give people a better immune response? And that might help with clearing the virus in a viral reservoir. Well, PD1 blockade actually in macaques actually seems to increase, as does CTLA4 blockade, seem to increase the immune response against HIV in macaques. And there's now a suite of these antibodies available. So there's ipilimumab, which blocks CTLA4. There's novalimumab that blocks PD1. There are others that block PDL1. And these have been approved for human use, but they've been approved for the use in various cancers that are end stage. And so these have revolutionized the treatment of end stage cancer with these sorts of cancers, particularly melanoma. The trouble is that although these cause very effective immune responses that are anti-cancer, they are associated with very significant toxicities that reflect the immune dysregulation that they cause. So they take the brakes off the immune system. The cancer gets killed, but at some cost to the patient. So they often get inflammatory colitis, they can get myocarditis, they can get pneumonitis, thyroiditis, they can have an autoimmune response that knocks off their pituitary. You get skin, autoimmune diseases in the skin and liver and in the CNS. So the side effects really mitigate their utility outside end-stage malignancy. And so unless you've got a patient with end-stage malignancy with HIV infection, these are really not effective drugs to use at the moment, though there are some clinical trials going forward with those, and we'll watch those. There are other drugs available that are effective modulators of the immune system, such as these derivatives of thalidomide, such as pomalidomide and lenalidomide. And they cause significant immunomodulation as well. Pomalidomide and lenalidomide increase the production of interferon gamma and IL-2, IL-10 and RANTES from T cells. They inhibit the function of T regulatory cells, and they enhance NK and NKT cell activity. So these have now been shown, or at least pomalidomide has been shown by Mark Polizzotto to be effective in allowing the clearance of Kaposi's sarcoma from people with resistant Kaposi's that has failed other therapies with minimal toxicity. And so we're currently exploring the role of these in pre-malignant HPV lesions. And so this may be a generation of, or immune regulators such as this, may be immune regulators that we can use to treat the morbidities associated with chronic HIV infection. But we'll need to watch that space, and perhaps I can present that next year if I get an invite. So in conclusion, immune regulation is complex, and so it's governed by a complex interaction of multiple factors. Not all interventions that you'd expect will work. I've told you several times during this talk that IL-2 is deficient from early on in HIV infection. We've replaced IL-2 clinically, and we know it doesn't work. So, understanding the complexity of these interactions is absolutely essential to the rational design of these therapies. So we need to understand these basic mechanisms so we can rationally design these interventions. And currently, my view is that the checkpoint inhibitors, although they're incredible in end-stage malignancy, are not appropriate for the use in HIV-infected people outside very well-regulated clinical trials. And we really need clean and more specific modulators of immune regulation. Thank you.
So, why there is regulation of the immune response؟ سؤال يطرح نفسه. تعرفون إذا كان الانفلاميشن مستمر after control of infection, there will be more destruction of cells and tissue. So, after complete removal of the antigen, invading antigen, there will be some mechanism that will certainly downregulate the immune response. The benefit of this is to decrease the collateral damage to the infected cells. At the same time, removal of all inflammatory byproduct in the microenvironment of the inflammation, like pus cells, repair the damaged cells, and so on. So, regulation in immune response to antigen has been previously described. Recognition of antigen in the absence of co-stimulation resulting in energy. Recognition of antigen with CTLA4 engagement of B7 resulting in downregulation of T cell activation, also for PD-1 molecules. Cytokines with stimulatory or inhibitory activities on immune system. Idiotype and anti-idiotype interaction leading to stimulation or inhibition of the immune response. Dose and route of antigen exposure can induce differential T response. Small dose of antigen may result in tolerance. High dose of antigen may result in effector function and vice versa, in which case can protect and in another can tolerize. كثير من عندنا أصيبوا في مرحلة ما في مرض معين. The best example تشوفون ال COVID-19 infection. In some cases there will be a mild infection, even asymptomatic infection, and in other families you see high morbidity and mortality in these families. So what is the explanation? As we said, the viral load is an important mechanism. Immune system of the body toward the invading organism, and another factors which is within the genetic constituent of the person himself may play a vital role in response to infection. So genetic factor influencing immunoregulation. MSC-linked gene help control response to infection. Certain HLA haplotypes are associated with individuals who are responder or run non-responder, those who are susceptible or resistant to infection. Also, non-MHC genes are also involved in immunoregulation. An example is a gene related to macrophage activity encoding transporter protein involved in the transport of nitric oxide into the phagolysosome. Natural resistance associated macrophage protein 1 polymorphism and this gene could change the activity of macrophage. Cytokines, chemokines, and their receptors are involved in immunoregulation as discussed previously. Polymorphism and gene encode these. In a particular, the receptors have been shown to correlate to susceptibility to infection or generation of an autoimmune disease. So these mechanisms will affect the response of individuals and the development of tolerance. So in summary, there should be a tight control and regulation of the process of inflammation and other diseases. Thank you for listening.